Hello, everyone, and welcome. This is Will Akey. Thank you for joining today's webinar. I'll just give a few moments here for others to join, and we'll kick off. Really appreciate you joining us today. We're really excited about today's content. All right. Again, hello and welcome. You've joined Informa's webinar series. Thanks for joining. This is Will Akey. I'm a product manager here with Informa and moderating today's session. We're joined by two esteemed colleagues here at Informa, which we'll introduce in just a moment. I did want to welcome everyone and thank you for joining. Certainly, today's exciting topic will be interesting for all of you that are participating today. I did also want to point out that we do have a series of webinars ongoing. This is, I believe, the second here at Informa. Next week's is on Trump's impact on healthcare and the opportunities and difficulties that people will have. That's next week, July 26th. So please keep an eye out for that if you're interested in participating in more of our webinars. Before we begin, I did just want to introduce ourselves. We're Pharma Intelligence, part of Informa, and have several products that service the healthcare market. Most notably in here and the ones that touch on benchmarking, which we'll touch on a little bit later, are Data Monitor, Healthcare, and Biomed Tracker. We have a long history of benchmarking in the pharma industry, and as well as our newest product, Pharmapremia, which is a new analytical tool that leverages a lot of our insights and data historically. And as always, we service our clients on the consulting end on the benchmark side. But we are a broad suite of products and I've worked with the industry for some time. Today's agenda is relatively straightforward and hopefully will be interesting for all of you. I'd like to just set expectations so you know what you're getting into as the hour progresses. Certainly the introduction to probability of success benchmarking is important. We want to set the stage for what we think of as success and failure. Our methodology is unique to our products and definitely want to make sure that you understand what we mean when we say success and failure. We'd like to dive into more oncology-specific benchmarking as a focus for today, how that compares to non-oncology disease groups as well as you break down oncology. And then we'd like to dive sort of a little bit more deeply into both some of the parameters that cause success or cause failure, as well as how top companies in oncology fare both compared to each other and based on their acquisitions and in-licensing activities. So as I mentioned, I'm joined by two colleagues here in Informa with a wealth of experience on both oncology and benchmarking. Firstly, I'd like to introduce Robert Jang. He's a senior scientific analyst with our Biomed Tracker product. He manages the scientific analyst team, specializes in inflammation and oncology, and has attended ASCO, I believe, for the past decade, so certainly has a wealth of insight on oncology and how people choose to go about it. So welcome, Robert.
Hey, everyone.
Thank you. And I'd also like to welcome Ellen Moore, who's a consulting director with Pharma Intelligence Consulting. She's worked as a consultant for over a decade, and most notably with Data Monitor Healthcare Consulting. She's worked with our clients to take our data within our data sets and to really drive insights to make business decisions and strategic decisions within healthcare. So we welcome Ellen as well. Hello, Ellen.
Hi, Will. Thanks. Hi, everybody. Thanks for joining.
And lastly, as I mentioned, I'm Will Akey. I'm the Senior Product Manager here at Informa Pharma Intelligence. I'm in charge of Pharmapremia, which, as I mentioned, is our newest tool. It drives insights and success rates as well as cycle durations based on our Biomed Tracker data. It's recently come to market and what a lot of our data in today's presentation is based on. If you would like more information, there's a link there, and we'll talk a little bit more about that as we go on. And as we progress, we hope to answer all of your questions, both on methodology and other questions that might come up. But if not, there is a chat box and a question box on your webinar, so please ask them as we go, and we'll try to answer them as we progress as well as at the end, and if not, certainly offline after the webinar is over. So what is our experience in benchmarking? Informa has published a study in Nature Biotechnology, which has been cited pretty well across the industry and driven a lot of questions to us. Michael Hay, our head of Pharma Intelligence Products, partnered with Bio and published a study on historical benchmarks over a decade. And it was pretty well cited and sort of expanded on the research that had been done in the industry on more of the scientific and publication side. And with that, we've used those insights to drive some of that information into our Biomed Tracker and Data Monitor Healthcare products. And since then, we've launched our newest tool, Pharmapremia, and also driven a lot of these insights into our consulting business. And we wanted to take a little bit of what our analysts are looking at in the market and take that to you today and tell you a little bit about how we're thinking about benchmarking and success. We have over 5 years of working with this data after this publication and definitely have found some interesting pieces as things in the market change. And we've chosen to focus on oncology today, both because it is the largest market, as well as because of the ever-changing aspects of oncology. So where are people using benchmarking? Certainly you all will know firsthand if you join this webinar why it's important to you, but some ways that we have found our customers use this is both in a portfolio evaluation and assessment of their pipeline. So what is my pipeline worth based on the phase that my products are in and how they might get to market, how fast they might get to market, and how much they might earn. And then conversely on business development decisions. So what is their pipeline worth? If I'm going to acquire a company, how much is that going to cost us based on what we think the future value based on success rates are? Additionally, peer comparisons and competitor comparisons. So if I'm a large pharma, are we really having more or less success with our strategic decision making than other competitors? Or if I'm not a large pharma, who should I compare myself to? And is our speed to market actually better than some of our larger competitors? And lastly, one thing that I think everyone that participates in any benchmarking activity is interested in is understanding the viability of our industry as a whole. Do we have enough in the pipeline based on success rates to continue to bring new and novel products to markets and patients? Do we have the ability to invest enough money to get the return? And so we have a chart there on the NASDAQ Biotechnology Index over the past decade, and you can see the valuation of the market as a whole fluctuates based on lots of things that go on in the industry, whether it's pricing and reimbursement actions or comments from politicians, or whether it's novel products coming to market in oncology. Certainly a lot of these parameters are ever changing, but understanding the historical success rates of products really drives the understanding of the industry as a whole and whether we really are sustainable. And with that, I'll turn it over to Robert, who will take you through a little bit about our methodology here at Informa and our benchmarking background. So I welcome Robert. Thank you.
Okay, thanks, Will. Thanks for that introduction to the concept of benchmarking. I'm going to go into a little bit more detail about the methodology that we use here for our own benchmarking, but I'm also going to try to raise some general issues that anyone doing his or her own benchmarking might consider. So first, let me start with some definitions, since there is some interchangeability and some unofficial terminology floating around out there. Probability of success, probability of technical success, phase success, etc. For us, those are all the same. They basically refer to the probability of a drug advancing from one clinical stage to the next. That is, from phase 1 to phase 2, from phase 2 to phase 3, and so on. So in Pharmapremia, our baseline probabilities are based on Biomarker's database of nearly 13,000 drugs, and our probabilities are basically straight objective calculations. So for any chosen set of drug programs, we simply count the number of programs that have advanced to the next clinical trial phase, divided by the total number of programs for which we know an outcome, either advanced or suspended. So here's a sample calculation shown at the bottom of this slide for the success rate of a hypothetical set of drug programs from phase 2 to phase 3. So we have 100 programs that pass through a phase 2 trial during this arbitrary 5-year time period. And 50 have advanced to phase 3 or to a phase 3 trial. The development of 20 were suspended, and 30 remain in phase 2 with no guidance one way or the other. So the probability of success or probability of technical success is just simply the 50 advancing drugs divided by the total of 70 drug programs for which we know an outcome, giving us 71% success rate. So that's our objective baseline, and it's pretty straightforward to calculate. But there are actually a lot of issues to consider before making PTS calculations. First, what are you going to define as a program? What counts as a single program? Is it one drug in one indication? Is it one drug for all of its indications? This is especially complicated by the rise of large basket trials, which are becoming so common in oncology. These are the types of programs for which, very early in development, a drug is evaluated in multiple tumor types in a single trial, and then advanced only in the promising tumor types. Somewhat relatedly, rather than advancing in a discrete, stepwise fashion, some of these phase 1 trials are simply expanding promising cohorts while ending the less promising ones. So a single trial functions more like multiple programs. So if a drug can hit on any tumor type, even if it fails many others, it could be recorded as a single success. Alternatively, if that drug fails the basket trial, does that count as one failure? Does that count as multiple failures? These are just some issues to consider in building your own PTS. Another factor to consider is positive bias. Since companies tend to release positive data while not always disclosing program suspensions, so some of the programs for which we don't know the outcome and exclude from our calculation could actually be suspended, and the company just hasn't released any information on that. That's always a caveat with any data set that relies on publicly released information like our database is. But even if you're doing this in-house, and you may have non-public information on your own company's failed programs that haven't been disclosed to the public, but you might not have that information on your competitors with whom you want to make a comparison. So again, positive bias is always there. Okay, real quickly, likelihood of approval. It's basically the same concept as PTS, but just focused on the probability of advancing from any phase all the way through to regulatory approval. So very simply, LOA is just the probability of advancing for each phase multiplied by the PTS or POS for the next transition. So in the example in the little red box, the LOA from phase 1 would simply be the phase success rate from phase 1 to phase 2 times the PTS for phase 2 to phase 3, and so on and so on. So you multiply all those phase success rates together to get an LOA of, in this case, 10.8%. You can do this starting in any phase. Just multiply the PTS for all subsequent transitions to your starting point. Okay, so enough with the methodology. If you're still with us, let's talk about something a little more interesting, like some actual numbers that we've come up with in Pharmapremia using the methodology that I just described in the Biomed Tracker database. So most of the data we're presenting today comes from a 10-year slice from 2006 to 2015, which includes just under 10,000 phase transitions and nearly 7,500 development programs. We'll start with the most general analysis, and then we'll work our way towards some oncology-specific data cuts. So for the broadest look, this slide shows the probability of success and LOA across all diseases in that 10-year slice. It's pretty apparent that the lowest success rate by far comes at the transition from phase 2 to phase 3. This probably isn't all that surprising. It makes a lot of sense since phase 2 trials are where most drugs get their first test of efficacy and where most go, where most go or no-go decisions would be made before committing to an expensive phase 3 pivotal trial. The graph also shows that the overall LOA for all drugs that enter clinical study through to approval is just under 10%. And that's an important baseline number as companies have been trying to justify the high cost of drugs by citing the low success rate of drug development. Okay, so now we'll start doing some narrower PTS analyses with a focus on oncology. Okay, so this graph shows the LOA from phase 1 for different therapeutic areas as categorized in Pharmapremia. You can see our 9.6 average LOA across all diseases from the previous slide shown here in purple. And at the left end of the graph, you'll see the therapeutic areas with the highest success rates, including hematology, for which, amazingly, nearly 1 in 4 drugs entering the clinic are approved. And to me, in some ways, this makes sense. It's not really that surprising since it includes hemophilia, anemia, blood protein deficiencies, diseases for which our biologic understanding is quite good, and the therapies are more or less straightforward, things like factor replacements, etc. At the right end of the graph, you'll see the below average therapeutic areas, including oncology at the very end with only a 5.1% phase 1 LOA. Also, you might have already guessed that the location of the 9.6% average in purple, that's way down on the right end of the graph, means that the N values or the sample size for the lower success therapeutic areas are very large. In fact, oncology and neurology had the 2 largest N values So they're really mostly responsible for dragging down the overall average LOA. To me, that's actually a really interesting finding, that companies are throwing up more chances in indications that have lower success rates. Honestly, I don't really know the explanation for that. I could offer a few guesses, like perhaps it's the lure of the enormous markets from a business perspective, especially for oncology and neurology too. Or at least for oncology, I could believe that there really just are more preclinical targets to pursue than in some other areas. I've worked as a cell biologist in my former life, and I know that it's relatively easy to come up with things that affect cell growth in a tissue culture dish. So there are a lot of preclinical targets to pursue, whereas in some other therapeutic areas or some other indications like, let's say, Alzheimer's disease, well, we're still really even debating what the biologic problem is, let alone finding good, viable targets. Okay, so let's move on. Oncology is the low performer by overall LOA. So here we break it down to the success rate of each phase to try to figure out where development is underperforming. This graph shows oncology probability of success for each phase transition in oncology versus all other therapeutic areas lumped together in the pink. And you can see that the phase 1 and the NDA or BLA success rates are pretty similar in oncology and outside of oncology. But what really stands out is oncology's low success rate at phase 2 and especially phase 3, where the 40.1% PTS is more than 20% lower than for other therapeutic areas. So let's expand on that, low phase 2 and phase 3 success rate. I actually didn't include a slide of a phase 2 breakout, but here's a breakdown of phase 3 rates across therapeutic areas. So unfortunately, for the phase 2 success rate breakout that I didn't include, interestingly, oncology was not actually the lowest specific therapeutic area when broken out that way anyway. Actually, psychiatry and cardiovascular indications were even lower. So it really looks like phase 3 is where oncology stands out as underperforming. In this case, a full 15% below the next lowest therapeutic area, cardiovascular. So again, to me, this might suggest that the early endpoints like response rate that are really common in phase 1 and 2 oncology trials might not really be that good at predicting more accepted regulatory endpoints like overall survival that take, of course, a long time to measure, so they aren't often incorporated into early trials. Another possibility, as I mentioned before, is simply the lure of the enormous oncology market that just might push companies to take risks on expensive phase 3 trials with even marginal earlier trial data. Okay, let's move on. So ironically, that shows that we have a low phase 3 success rate in oncology, but ironically, the FDA has been quite lenient for oncology development, which provides yet another incentive for companies to go forward. Here are the success rates after regulatory filing, and oncology is actually the highest after first review at 79%, up in the top of the first column. The ultimate approval rate after multiple rounds of review isn't the highest in oncology, but it's still close to the average across all therapeutic areas. So I said lenient, but really, one explanation for the high first round review success rate could just be that the FDA is quite clear and consistent on its requirements in many oncology indications. So companies know exactly if a filing will be acceptable or not based on their clinical trial data. Also, I have felt that the FDA has become more lenient in the last few years, especially in oncology, where there's been some controversial and high-profile approvals based on really a marginal clinical benefit. A few more analyses in my last few slides just to show what's possible and how we can cut the data more and more narrowly. So here in this slide, we've taken that low phase 3 success rate in oncology and broken it down by individual indications as classified in Pharmapremia. You can see that the hematologic cancers at the top in purple are generally more successful than for solid tumors at the bottom in pink. And there are a few solid tumor types that really have quite low success rates, namely pancreatic, gastric, and ovarian. And again, this sort of makes sense. We know that our measures for hematologic cancers are quite strong, whereas some of the early work or the preclinical work with solid tumors might not translate to more difficult to treat solid tumors when we have to consider the tumor microenvironment, et cetera. Okay, my last two slides, just show ways to incorporate other variables into a PTS analysis. Just note that I borrowed these slides from Ellen, so note that they aren't exactly the same data set as the previous slides, but they still illustrate the point. So here we've added a variable of molecule type, comparing small molecules to biologics. And we found that biologics actually fare better at every phase of development. So incidentally, just to take you through this slide, the numbers in the brackets and in the parentheses are actually just the number of drugs and the number of programs, respectively, in each group on that graph. Okay, my last slide here is just adds a biomarker variable where we compared the success rates with and without some biomarker in the trial design. Perhaps not surprisingly, we see that programs, including some refining biomarker, shown in purple, fared better at every stage of development. And this was true for solid tumors in the top panel, as well as heme cancers in the bottom panel. In some cases, the differences were really substantial, emphasizing the benefit of having a biomarker to focus the population most likely to derive a benefit from any particular drug. So that's really a little tour of oncology and an example of some of the ways that we can incorporate, or anyone can incorporate, different variables into a specific PTS analysis to be most relevant for any goal. Now I'm going to turn it over to Ellen, who's going to take us through more of a company-focused look at success rates. Ellen, I'll turn it over to you.
Awesome. Thanks, Robert. Thanks. And thanks for crediting me with those slides. I do think it's important to point out that there were some minor differences in the timelines for which that data was pulled. So I can briefly let everybody know some details about the slides I'm going to share. I have a particular interest in looking at company-specific analysis. I spend a good deal of my time working with sell-side companies and valuing their assets and evaluating strategic partners. And so this plays a role there with regards to assessing the extent to which a company can be confident in structuring the deal with a great deal of royalty rates versus upfront payments. So let's talk about the select companies that I'm going to show you in a little bit of detail. The companies that I have analyzed and will term in the following slides as, quote, select companies are those that fall into the top 20 pharma companies by total market cap and have a significant number of oncology phase transitions from phase 2 onward. So those companies are Roche, Novartis, Eli Lilly, Pfizer, AstraZeneca, Sanofi, Bristol Myers Squibb, Amgen, Celgene, AbbVie, and Merck. One company that does fall into the top 20 by total market cap and also has a large number of oncology transitions that's not included is Teva. And the reason they've been excluded is because so many of their phase transitions fall in phase 1, and they didn't really have a substantial subset in phase 2 or phase 3 over the 5-year time period of 2011 to 2015. So we're looking at a sum total of 11 companies included in our select company analysis that I'm going to take you through right now, and we will compare that to the rest of the industry. Here I highlight the hematology and solid tumor numbers that Robert has already taken you through We also split it into the rates for the combined select companies and all others in the marketplace. Across all companies taken together, as Robert pointed out, the programs targeting hematologic tumors have over double the phase 2 likelihood of approval of solid tumors. So that is this 12% to 5% in the phase 2 LOA column. This is driven largely by a significantly greater phase 3 probability of success. When you look at this phase 3, probability of success is 54% to 32% when you're comparing heme to solids. So when we look at the select companies and make the same comparison, those companies are only marginally superior to the rest of the industry in their phase 2 LOAs. So again, their phase 2 LOAs are 14% for hematology and 8% for solid tumors, versus other companies that are at 11% for hematology and 3% for solid tumors. In solid tumors, they do achieve a pretty significant phase 2 LOA in comparison to the rest of the market. We're looking at 8% there and comparing that to 3% for other companies. So these are all single digits, but that's a pretty big span. For solid tumors, the superior phase 2 LOA achieved by select companies, you can see, is driven by the higher probability of success in phase 3 and at the NDA level. So again, that's here. Phase 3 being 39% probability of success for solid tumors in phase 3, and on 93% at the NDA phase. And that's in comparison to 23% phase success for solid tumors for all other companies and a 67% NDA phase. So this select group of companies is markedly better in later stages of development and regulatory filing than its competitors in smaller, less notable companies. It's also interesting to point out that these select companies have a significantly greater success total at the NDA stage than the rest of the companies. So that's 91% in all oncology when you combine heme and solid tumors versus 76% for all other companies. Breaking it out by company provides a little bit more detail and a little bit more data of interest. So I'm going to focus us on just a few companies, and you're probably going to have to squint a little bit. I'm not actually able to work well this mouse, so I'll point out the numbers of interest and let you hone in on them. There are wide differences in phase 2 likelihood of approval numbers across the cohort that are immediately apparent. So for phase 2 LOA, you're looking at AbbVie with a 26% likelihood of approval from phase 2 in oncology, and comparing that with, say, Sanofi, which is barely registering above 0 at the bottom of the list. So that's a huge span in phase 2 likelihood of approval numbers, which, I'll just reiterate, is a compounded factor of their phase 2 success rates along with phase 3 and regulatory filing rates. So interpretation of any individual LOA requires consideration of these two key factors. So first is the number of programs contributing to their success rate, and the second is the number of drugs on which those programs are based. So you're going to see me highlighting that in some of these charts, both the number of drugs included in the analysis as well as programs, where we're defining programs as a drug and indication combination. So any drug with any one tumor type would be a program. For example, AbbVie's phase 2 likelihood of approval is driven by an exceptionally high phase 3 probability of success. So that's 80% probability of phase success at phase 3 But this is also based on a relatively low number of contributing programs. So I'll point out AbbVie's successes specifically in a further slide, but I want to contrast this with Roche's 14% likelihood of approval at phase 2, which is nearly half that of AbbVie's, but it's based on a considerably greater number of phase 3 and regulatory transitions. So this increases our confidence that Roche is able to repeatedly innovate at the same rate above the average for select companies, but not quite matching that of companies taking more of an opportunistic approach to their oncology portfolios. When we look specifically at solid tumors, I apologize for all these data tables, but they were a quick and easy way of showing you multiple numbers at once, and we will get into some more easily digestible charts in a minute. Looking strictly at solid tumors, the differences between companies and the breadth of their portfolios becomes starkly evident in the analysis of solid tumor success rates for the 5-year period that we analyzed. So Merck's phase 2 likelihood of approval is double that of most of its competitors. You can see it's at 22%. But this derives predominantly from dramatic success of the development of just one drug, Keytruda, and multiple indications. So this narrow focus can play in the opposite direction as well. Lilly's dismal phase 2 LOA, which is at 2%, despite being based on a large number of phase transitions, is largely attributable to just 2 drugs. Those were ultimately suspended from multiple indications simultaneously. So I'll show those examples in a minute as well. The difference between the outcomes for Merck and Lilly emphasizes the gamble taken when a company focuses its R&D portfolio narrowly with regard to the number of drugs, but broadly with regard to the number of indications. So clearly the prospect of multiple wins from a single product is enticing, but the loss to the R&D portfolio if a drug fails across the board can be substantial and have a significant impact on these particular figures. One more notable thing of mention is Roche's phase 2 LOA, which is second only to Merck, but nonetheless considerably lower. That number in this slide is 14%, is based on roughly triple the number of programs, stemming from 4-fold the number of drugs. So those rates that you're looking at here for Roche result in a whopping number of 8 approvals over the time period. So let's look a little bit more specifically at Roche. Before I start talking about this particular chart, I want to explain it a bit. I use this table to depict the successes and failures that Roche saw over the 5-year time period that we analyzed. So for programs like Avastin in ovarian cancer, Avastin in cervical cancer, Perjeta in breast cancer, those programs likely had phase 1 and phase 2 trial data, but that data and the transition into phase 3 was not captured in this particular data period. So you'll see something like Cotellic for melanoma made its way from phase 1 through to regulatory approval within that 5-year time period. The same successes were likely seen with those programs that don't have a color in the earlier phases. It's not there simply because it didn't take place during the time period that we analyzed, that being 2011 to 2015. So let's get to discussing this data just a bit. What's not captured on this table is all of Roche's phase 1 activity. In total, over the 5-year period, Roche had 99 programs in development from 47 drugs. So they were the most active developer of oncology drugs during the 2011 to 2015 time period. They had a 100% regulatory success rate, leading to 9 approvals from 8 drugs across 5 indications. 6 of their approvals were lead indications for those drugs. Alectinib was notable for approval within 5 months of filing the NDA. So Aravej also was granted accelerated approval based on phase 2 results. So this is just trying to give you a visual of the data that we're looking at and how we are calculating success rates. So these red suspensions are considered failures, essentially, for the purposes of our calculating the phase 3 success rates, whereas alectinib, Cotellic, Avastin for ovarian and cervical cancer, Perjeta, and Zelboraf were all considered successes at phase 3. Looking at another company to give you a little bit more flavor for how the analysis can be understood is Novartis. So they had productivity roughly half that of Roche over this time period, but still the second most active developer in solid tumor indications. So they had 49 programs total over the 5-year period from 24 separate drugs. Similar late-stage activity to Roche, but much lower activity in phase 1. Their lone regulatory failure was Zometa for breast cancer. So Novartis withdrew that application due to follow-on trial results that became available after the NDA filing. 3 of their 7 approvals were granted to drugs which had previously been approved for other indications. So this is a bit in contrast to the lead indications and 6 drugs that Roche had approved over the same time period. Their remaining 4 indications were, 4 approvals were for lead indications, so still a significant amount of new drugs to market over the 5-year period. They also had a drug with accelerated approval, that being Zicadia, on phase 1 data, and that was granted within 3 years of them entering the clinic. So you can see actually that Zicadia had a pretty rushed approval, with all stages of development being completed in this 5-year time period. Merck is another company worth analyzing for the sake of understanding what's going on in the solid tumor space. In a total of 24 programs from 11 drugs, they saw their successes characterized centrally with the successes and failures of Keytruda. So all of Merck's successes from phase 2 onwards, with the exception of Pegantron for melanoma, are related to Keytruda. Counterbalancing their broad success with Keytruda, Delatuzumab accounted for 80% of Merck's phase 2 failures. So this is one more example of how developing one drug across multiple indications can have a significant impact on the rates that are calculated for the probability of success. I've taken up a lot of time with this solid tumor stuff, so I've got some AstraZeneca data here, but I'm going to skip over it to move on to hematology and keep things moving a little bit. Back to tables, but again, there's some interesting findings here. Amgen and AbbVie led in late-stage development for drugs for hematologic tumors, both having 100% success rates from phase 3 during the 2011 to 2015 time period. The number of transition on which Amgen's and AbbVie's late-stage success rates are based is low, but that's true across all selected companies. So no company had more than 4 transitions at phase 3 in hematologic tumors over the 5-year time period. Of the companies with 4 transitions at phase 3, Pfizer had the lowest success rate, so less than half of the select company average, and markedly lower than its phase 3 success rate in solid tumors. Pfizer also had a below average success rate at phase 2. Celgene, AbbVie, and Amgen stand out for impressively high phase 2 success rates. And in AbbVie's case, this is based on one of the highest numbers of transitions, that being 10. In contrast, Sanofi's 11 phase 2 transitions have led to the lowest success rate among selected company cohorts. And that was at 0%, again, at phase 2. You can see, actually, that Sanofi just has very little going on in the hematologic space. So that noticeable 0% at phase 2 is based on very little activity in the space to begin with. I'm going to give some additional company examples here, and I hope this is of interest to you. Amgen, for hematologic tumors, is seeing successes with Kyprolis and Blincyto, and both were given accelerated approval, and Blincyto was granted breakthrough designation 5 ahead of its PDUFA date. If we look at AbbVie's hematologic tumor successes, you can see a great deal of this is reliant on Imbruvica. What's important to note with our data set is that while Imbruvica was developed by Pharmacyclics and its partner, once the acquisition of Pharmacyclics by AbbVie takes place, we consider those programs to be AbbVie successes or failures. So those numbers account for a great deal of their successes and their very high likelihood of approval numbers. The AbbVie's total development investments over the 5-year period included 13 programs from 6 separate drugs. As I said, there was heavy reliance on Imbruvica, responsible for 6 of the 7 late-stage successes that they had. Its approval in mantle cell lymphoma was the lead indication, and the drug was granted accelerated approval in both MCL and CLL. So you can see a great deal of movement, both from phase 1 all the way through to regulatory approval with Imbruvica in multiple indications. When we look at Roche, there's a very significant amount of development in hematologic tumors as well as there was in solid tumors. So 21 total programs from 15 drugs over the 5-year time period. Gazyva accounts for all of Roche's late-stage successes in the hematologic arena. Its approval in CLL was the lead indication. And elsewhere in its hematologic pipeline, Roche was hit hard by phase 2 failures for Panitumumab, Tarceva, and Arivedge. This analysis was particularly interesting to me. There's not a lot very telling when we look at Pfizer's acquisition of Medivation versus what the acquisition would have done to Sanofi's success rates. But I found it interesting to do the analysis nonetheless. You can see that the pink bar being the Pfizer numbers post the inclusion of Medivation successes and failures in comparison to the purplish bar, which was Pfizer's success rates without the inclusion of Medivation's transitions. There's not a heck of a lot of difference in the phase success rates in any one phase. However, if you look at what an acquisition of Medivation would have done to Sanofi's success rates, it's a little bit of a different story. I'm sure everybody understands that Sanofi was in the game with regards to a Medivation acquisition, and this just gives you a snapshot of what the acquisition would have done to their success rates. Now, clearly, probability of success rates are not the driving factor behind acquisitions, but I find it interesting given the benchmarks of clinical development success rates to see what impact an acquisition has on any one company's successes. I want to contrast the Pfizer Medivation success rates with the success rates for AbbVie, both pre and post the Pharmacyclics acquisition. So the purple bars represent AbbVie without the inclusion of Pharmacyclics transitions, and the pink bar is AbbVie and Pharmacyclics success rates combined. So here you can see a huge difference in the success rates over time. In this case, the acquisition had a large impact on the success rates in all phases and a greater impact on the size of the overall portfolio in all phases. So huge difference in sample sizes as well as actual success rates. I don't have other slides to share at the moment on this on these company-specific rates, but would be happy to answer questions as they come up and conduct additional webinars if there were specific cuts of data that were company-specific and of interest to any of you listeners. So, I will hand it back over to you at this point to sort of tie things up.
Yeah, great. Thank you, Ellen. I certainly find the acquisition analysis very interesting, so appreciate that. And so I just wanted to kind of summarize what we talked about today and give some follow-up for all of you. Certainly benchmarking is an important exercise across our division. We do feel like we have the ability and insights to drive some of these decisions from the macro level that Robert went through all the way down to the micro level that Ellen took us through on a company-by-company basis. Certainly it gives you new insights into the drug development process and how companies choose to augment their pipelines. You can see that there's a wide variability across diseases, drug classifications, companies, and many more parameters. So definitely something to keep an eye out, and you can tell almost any story with the data depending on what you're looking to accomplish. I think Ellen's slide certainly showed that acquisitions can change everything, and exactly how they impact success rates is very interesting to look at, to see exactly how perception can change in the marketplace based on one key deal over 5 years that we saw there. And again, to change internal perception, I think internally here at Informa, we frequently use this data to drive discussions further when we're talking about possible success rates of drugs that we follow in our databases and products. And certainly it can do that for you and your company as well. Certainly having the underlying data allows you to do that. Robert touched on a little bit about the positive and negative biases of our data set, but ultimately having the information and what is driving these successes, as Ellen touched on in her company-by-company analysis, helps decide whether the perception is reality. And as Ellen just mentioned, certainly we're here to help. We have both a product solution for benchmarking at a high level in Pharmapremia, as well as the more granular cuts down to the company level, as Ellen touched on through our consulting division. And lastly, I wanted to thank you all and our presenters, both Robert and Ellen. Really appreciate their insights and expertise on this matter. And thank you all for joining us as well. If you're interested in more follow-up, please reach out to any of us at premiasupport@informa.com or through your account manager or through our websites. We definitely are here to help and follow up with any questions as well. So with that ends the content portion. We do have a couple questions that we can take and answer here. If you have any more, please feel free to type them into the chat box. The first one that I'll take is just on the slide that Ellen presented that showed some fairly small n values on the company cuts of data. And how does that really tell us and inform us the ultimate probability of success, given that the statistical nature suggests that has a lot of variability? And what we've worked on with some of our customers, and especially Pharmapremia, is to use proxies for the data that you're looking at, or expand your time frame, or expand your parameters. And as you start to take steps back and include more programs, the numbers become more telling. And sometimes the information you include becomes a little bit biased, but you need to understand why that is. So one example would be obesity. Before 3 products were approved in obesity in 2012, there really hadn't been anything approved in obesity for some time. And what we chose to do is take several layers of endocrine diseases and look at things that had success and where those happened and where they didn't happen, and how that might inform the latest developments in obesity. Additionally, you can look at things like a novel biologic versus the rest of the field, or an orphan disease. You can take other orphan diseases and use that as a proxy as well. So you need to take multiple cuts of data to almost triangulate a better PTS number, whereas when you get to that more granular information that Ellen showed, you get the true value. And the true value is just as important to know, are you coming even close to that proxy that you've used? The next question, which I believe Robert's probably most able to answer, is specific to the early discussion on basket trials, and how do we look at a typical oncology trial that's more of a phase 1-2 trial. So how do you go from that all-comers into the maximum tolerated dose within a specific indication or population? How do you treat that product or that trial say success? So Robert, do you want to take that?
Sure. So if I understand this question correctly, or what I think this question is asking, is something that we touched on earlier. How do we take these basket trials that address multiple tumor types, or that address multiple tumor types, and then eventually keep expanding or lead to development in specific indications? And, you know, as I was suggesting earlier, this is a judgment call. And we use our judgment here at Pharmapremia and Biomed Tracker. So we can, typically what we'll do is, if a single umbrella trial, you know, covers multiple tumor types, it might be listed as a single trial under a generic indication like solid tumors. And then as development proceeds farther in specific indications, we'll essentially split or multiply that trial. So if, you know, it's all comers in a phase 1 trial, but then eventually development proceeds in 2 tumor types, we'll consider that 2 programs and 2 successful phase transitions. This is a judgment call, but we also have the ability to retroactively change things in the database. So we have the benefit of seeing where companies have developed something. So after they've moved to phase 2 or phase 3, we can look back and say, okay, how did development proceed? And we can fix that in the database. So I hope that answers that question.
Yeah, thank you, Robert. Another question here is, do we make a distinction between suspensions due to specific parameters, whether it's technical failure, safety, efficacy, et cetera? So certainly when we do these more granular assessments, we do try to parse out that distinction if that's of note. But as Robert mentioned a little bit in the bias in public reporting, a lot of times this information on the suspension side isn't really very well articulated. So a company may have some information on a product in their pipeline at every quarterly earnings conference call or on their company website for some time, and then eventually just drop that product or program off of that slide presentation. And so our analyst team scours the public information and public domain for whether there's any ongoing trials that might actually lead to success and bring it sort of back to the company's forefront, or whether there's any other presentations that have happened at any medical conferences that would suggest the trial was successful or had failed. But a lot of times that information just isn't well known. So we don't make the distinction in our products at this point on the failure analysis, just because of the biased nature of how it's reported. So oftentimes the company will say it's a quote-unquote business decision or a strategic decision, when that really just means that the efficacy wasn't there or the safety wasn't there compared to the products that are on the market. So it's something that we can do, especially for some of the products, as it is often reported through trial data, or we can make an inference. But on the whole, we don't tend to do it at the macro level just because of that biased nature of the reporting. And as the last question or more comment is just on including more biomarker analyses and more specific targeted therapies, how that impacts the analysis. It's something that we've started to do and are able to do in more real time than other benchmarking endeavors that take smaller data sets. So certainly the success rates of, say, immuno-oncology assets or breakthrough designation are 2 that we were at the forefront of capturing and evaluating, because we see those things happen in real time and cover the entire disease market. As we continue to see more nuances in drug development and we're able to classify those programs, we will be able to have the likelihood of approvals parsed out by that information. I think the one thing that we caution is that as these things start to happen, again with the positive reporting bias, these things tend to show just successes in the early days, and the things that have failed don't yet yield their failures. So breakthrough designation is a great example where there's some, over 100 products now with breakthrough designation, but many of the ones that will ultimately fail just haven't reported their failures. So inherently they aren't calculated in the probability of success, and therefore it looks like all breakthrough products will be approved based on historical data. But really that's just a function of the fact that the ones that have been successful have already happened and we haven't heard the failures yet. So we do need a bit of a time lag in order to really truly evaluate it, but definitely we can parse the information out as we begin to classify new information like those included. And with that, that appears to be all of our questions today. So I really do want to thank everyone for joining us and for sticking it out through the end here. We really appreciate your interest and are happy to continue to provide benchmarking analyses for you through any medium possible. So please keep an eye out for future webinars, other content we produce, or reach out directly. Thanks again and have a great day. Bye-bye.